Cancer cells are notorious for their rapid growth and proliferation. To fuel this aggressive behavior, they require a constant supply of energy. This energy comes primarily from glucose, a simple sugar that our bodies break down from the food we eat. Researchers have discovered that cancer cells often have a unique way of metabolizing glucose, different from normal cells. This altered metabolism, known as the Warburg effect, has become a promising target for new cancer therapies. By targeting the way cancer cells produce energy, we can potentially inhibit their growth and survival. This approach is particularly exciting because it focuses on a fundamental vulnerability of cancer cells, offering a new avenue for treatment. Traditional cancer treatments, such as chemotherapy and radiation, often come with significant side effects. These side effects arise because these treatments can also damage healthy cells in the process of targeting cancer cells. This new approach of targeting cancer metabolism holds the potential for more selective and less toxic treatment options. By exploiting the metabolic differences between cancer cells and healthy cells, researchers aim to develop therapies that specifically target and eliminate cancer cells while sparing healthy tissues. This targeted approach could revolutionize cancer treatment by improving patient outcomes and reducing the burden of side effects. To understand how targeting cancer metabolism works, it's essential to understand the two primary ways cells generate energy: glycolysis and oxidative phosphorylation, or OXPHOS. Glycolysis is a process that occurs in the cytoplasm of the cell, breaking down glucose into pyruvate. This process generates a small amount of ATP, the energy currency of cells, even in the absence of oxygen. OXPHOS, on the other hand, takes place in the mitochondria, often referred to as the powerhouses of cells. This process is much more efficient than glycolysis, producing a significantly larger amount of ATP from glucose in the presence of oxygen. Normal cells typically rely on OXPHOS as their primary energy source, switching to glycolysis only under conditions of low oxygen. However, cancer cells often exhibit a preference for glycolysis even when oxygen is plentiful. This phenomenon, known as the Warburg effect, provides cancer cells with several advantages. Firstly, it allows them to survive and proliferate in environments with limited oxygen supply, such as within a growing tumor. Secondly, the byproducts of glycolysis can be used by cancer cells to build the building blocks they need for rapid growth and division. The Warburg effect is a hallmark of cancer and a key target for metabolic therapies.
Two promising drugs, 2-deoxyglucose, or 2-DG, and Fenofibrate, have emerged as potential weapons in the fight against cancer by targeting these energy-producing pathways.
2-DG is a glucose analog, meaning it has a similar structure to glucose but cannot be fully metabolized by cells. This characteristic allows 2-DG to act as a glycolysis inhibitor. When 2-DG enters a cancer cell, it competes with glucose for entry into the glycolytic pathway. Once inside the cell, 2-DG gets phosphorylated but cannot be further metabolized, effectively blocking the glycolytic pathway. This blockage starves the cancer cells of energy and inhibits their growth.
Fenofibrate, on the other hand, is a drug traditionally used to lower cholesterol levels. However, recent research has revealed its potential as an OXPHOS inhibitor. Fenofibrate exerts its anti-cancer effects by disrupting the function of complex Y in the electron transport chain, a crucial component of OXPHOS. This disruption leads to a decrease in ATP production, effectively cutting off the cancer cell's primary energy source. Moreover, fenofibrate has been shown to induce oxidative stress in cancer cells, further contributing to their demise. The ability of fenofibrate to target both OXPHOS and induce oxidative stress makes it a promising candidate for cancer treatment.
Recent studies have explored the potential of combining 2-DG and fenofibrate as a cancer treatment strategy, and the results have been promising. These studies have demonstrated that the combination of these two drugs can effectively inhibit the growth of various cancer cell lines in laboratory settings.
For instance, a study published in the journal Cancer Letters found that the combination of 2-DG and fenofibrate significantly reduced the viability of human breast cancer cells. This study also showed that the combination therapy induced apoptosis, or programmed cell death in the cancer cells. These findings suggest that combining 2DG and fenofibrate could be a powerful strategy for combating cancer.
Furthermore, the effectiveness of this combination therapy has also been observed in animal models. In a study published in the journal PLOS One, researchers found that the combination of 2DG and fenofibrate significantly inhibited tumor growth in mice with implanted human lung cancer cells. This study also reported that the combination therapy was well tolerated by the mice, with no significant side effects observed. These promising results from both in vitro and in vivo studies provide strong evidence for the potential of 2DG and fenofibrate combination therapy as a novel approach to cancer treatment.
These findings are really exciting and suggest that we might be on the verge of a breakthrough in cancer treatment. The combination of 2DG and fenofibrate holds great promise for the future.